Human chromosomes, general characters. Chromosomes are the carriers of genetic informations, as they contain the genes which are the units of heredity. Chromosomes are formed of macromolecules of DNA in the form of two strands twisted on each other to form a double helix. Human somatic cells contain diploid number of chromosomes, that is 46 chromosomes, of which 44 are autosomes and 2 sex chromosomes. Male cells contain XY sex chromosomes, while female cells contain XX sex chromosomes. Structure. Each chromosome is made of two chromatids connected at the centromere. Chromatids are made of units called chromonimata, embedded in the chromosomal matrix. Types. Chromosomes are classified according to the position of the centromere and the length of the arms into metacentric chromosomes. The centromere is centrally located. It may be with short or long arms. Submetacentric chromosomes, the centromere is found near the center. Also, it may have short or long arms. Acrocentric chromosomes, the centromere is found near one end. Also, it may be with short or long arms. Telocentric chromosomes, the centromere is located at one end. It is not present in human cells. Centromere divides the chromosomes into two arms, a short arm P and a long arm Q. Karyotyping. Definition. It is the method used to study and classify chromosomes. Technique. Leukocytes are the most convenient. Leukocytes are separated after centrifugation of a hyperinized blood sample. They are stimulated to divide in culture by adding a mitotic agent. The culture is incubated at 37 degrees centigrade for 72 hours, after which colchicine is added to the culture to stop the cell division at the metaphase stage. A hypotonic solution is added to the culture to swell the cells and to help dispersion of chromosomes. Samples are spread on slides, fixed, stained, and examined. Chromosomes are photographed and arranged according to the length and the position of their centromeres. Classification of chromosomes in human karyotype preparations. Chromosomes are classified into 22 pairs of autosomes, arranged in 7 groups, and 1 pair of sex chromosomes, according to the position of centromeres and the length of the arms. Recently, classification is based on the banding patterns, which are the different bands observed on the individual chromosome. Group A includes 3 pairs of long metacentric chromosomes, chromosomes of pair 1, 2, and 3. Group B includes 2 pairs of long submetacentric chromosomes, chromosomes of pair 4 and 5. Group C includes 7 pairs of medium-sized submetacentric chromosomes, chromosomes of pair 6, 7, 8, 9, 10, 11, and 12. Group D includes 3 pairs of medium-sized acrocentric chromosomes, chromosomes of pair 13, 14, and 15. Group E includes 3 pairs of short submetacentric chromosomes, chromosomes of pair 16, 17, and 18. Group F includes 2 pairs of short metacentric chromosomes, chromosomes of pair 19 and 20. Group G includes 2 pairs of short acrocentric chromosomes, chromosome 21 and 22. 6 chromosomes are XY in males and XX in females. X chromosome is longer than Y chromosome. Summary of karyotyping. The 7 groups of chromosomes are easily remembered as they are the first 7 letters of APC. So they are Group A, Group B, Group C, Group D, Group E, Group F, and Group G. The number of chromosomes in each group is respectively 3, 2, 7, 3, 3, and 2, 2. That is 3 pairs in Group A, 2 pairs in Group B, 7 pairs in Group C, 3 pairs in Group D, 3 pairs in Group E, 2 pairs in Group F, and 2 pairs in Group G. Chromosomes are grouped according to the type S. Groups A and F contain metacentric chromosomes. Group A contains long, while Group F contains short metacentric chromosomes. Groups P, C, and E contain submetacentric chromosomes. Group P contains long, Group C contains medium-sized, while Group E contains short submetacentric chromosomes. Groups D and G contain acrocentric chromosomes. Group D contains medium-sized, while Group G contains short acrocentric chromosomes. Banding techniques. It is the use of characteristics pattern of transverse bands to distinguish, study, and analyze the individual chromosomes. G banding is the most common, widely used banding technique, in which chromosomes are treated with trypsin and then stained with giemsa. Stain. Each chromosome is stained with characteristic light band containing more transcribed DNA, that is active DNA, and dark band containing late replicating DNA, that is inactive DNA. It was found that homologous chromosomes contain identical bands, which were given numbers according to the International System of Human Chromosomes nomenclature, ISHCN. Both short arms P and long arms Q are divided into regions, then bands and subbands, which are given numbers moving outwards from the centromere. The number of chromosome is written first, then the number of the arm, then the region number, and lastly the band number. An example: 4q24 means chromosome number 4, long arm, Q, region number 2, and band number 4. Clinical importance of karyotyping. Discovering abnormalities in chromosomes helps the diagnosis and treatment of different diseases. It helps the diagnosis of congenital diseases as Down syndrome, mongolism. It helps the determination of the sex of the fetus. It helps the diagnosis of primary amenorrhea, repeated abortion, and sterility. Also, it helps the diagnosis of certain diseases as chronic myeloid leukemia. Summary of karyotyping. The 7 groups of chromosomes are easily remembered as they are the first 7 letters of APC. So they are Group A, Group B, Group C, Group D, Group E, Group F, and Group G. The number of chromosomes in each group is respectively 3, 2, 7, 3, 3, and 2, 2. That is 3 pairs in Group A, 2 pairs in Group B, 7 pairs in Group C, 3 pairs in Group D, 3 pairs in Group E, 2 pairs in Group F, and 2 pairs in Group G. Chromosomes are grouped according to the type S. Groups A and F contain metacentric chromosomes. Group A contains long, while Group F contains short metacentric chromosomes. Groups B, C, and E contain submetacentric chromosomes. Group B contains long, Group C contains medium-sized, while Group E contains short submetacentric chromosomes. Groups D and G contain acrocentric chromosomes. Group D contains medium-sized, while Group G contains short acrocentric chromosomes. Chromosomal abnormalities. Definition. It is the disturbance in chromosomal structure and number. Types. Abnormalities of structure. These are changes in morphology of chromosomes. Deletion is loss or detachment of a part of one or more of chromosomes. Breaks of chromosomes are very common, as chromatid ends are sticky from electrochemical forces. Most of these breaks heal rapidly and correctly. Ring formation results from breaks of the distal ends of the same chromosome and their reunion in a wrong way to form a ring. Inversion is the wrong reattachment of broken segments of chromosomes. Segment from one arm is reattached to the other arm in a wrong way. Translocation is the transfer of segments between chromosomes. Segment from one chromosome is reattached to the other in the wrong way. Isochromosomes is abnormal transverse division of the centromere, separating the two arms, instead of longitudinal division separating individual chromatids. Abnormalities of number. These are changes in number by addition or loss of one or more of chromosomes. Polyploidy is abnormal duplication of haploid number of chromosomes. Normally, the number of chromosomes in somatic cell is double the haploid number, 2n. In triploid, it is 3 times the haploid number, 3n. In tetraploidy, it is 4 times the haploid number, 4n. These numerical aberrations are incompatible with life, impossible the life to continue, and cause miscarriage, that is, loss of pregnancy. Aneuploidy
is the addition, trisomy, or loss, monosomy, of a chromosome and may affect either autosomes or sex chromosomes. Autosomes, trisomy, is the presence of an additional chromosome to the usual pairs. It has been reported in all autosomes except chromosome number 1. The commonest trisomy is in chromosome 21, which means the presence of 3 of chromosomes 21. This is in Down's syndrome or Mongolism. Monosomy is the missing of one chromosome from the usual pairs, and monosomy of autosomes is lethal. That is impossible with life. Sex chromosomes, trisomy, is the presence of an additional sex chromosome, usually X. Normally, males have X and Y sex chromosomes and need both for sex differentiations, while females possess double X chromosomes. One of them is inactive, called sex chromatin or Barr body, and so females are positive for sex chromatins, while males are negative. Klinefelter syndrome, in this disease, males possess an extra sex chromosome that is positive for sex chromatin, and such males have differentiation or poor musculature and infertility and liable for breast cancer. Monosomy of X chromosome is detected in females. Females possesses 45 chromosome, 44 autosome, and 1 X chromosome. This is present in Turner syndrome. Turner syndrome is characterized by abnormality where 1 X chromosome is missing. The Barr body is absent. The disease of females is characterized by primary amenorrhea, underdeveloped genitalia and secondary sex characters, infertility, together with renal and cardiac abnormalities. Causes of chromosomal abnormalities: hereditary in the family, old age of pregnant women, infection with German measles during pregnancy, exposure of pregnant women to radiation or autonomic diseases.
Sex chromatin or Barr body. General characteristics. The sex chromatin represents an X chromosome which remains inactive, that is, coiled during the interphase. It is stained with basic dyes, hematoxylin, and is Fulgen positive. It occurs in females as their cells possess double X chromosomes, while males have Y and X sex chromosomes and need both for sex differentiation. As sperms contain 22 autosomes and either Y or X sex chromosome, while the ova contains always 22 autosomes and an X sex chromosome. Site: somatic female cells, as those lining the internal surface of the cheek, show Barr body as a darkly stained body or granule at the periphery of their nuclei, attached to the nuclear envelope. In neutrophils, Barr body appears as a drumstick-like structure attached to its nucleus. Clinical importance of Barr body: identification of the sex of the fetus and diagnosis of hermaphrodism, diagnosis of sex chromosomes abnormalities as in Turner's syndrome, diagnosis of the causes of primary amenorrhea, repeated abortion, and sterility. It is also important in forensic medicine and Olympic Games.